Hi everyone, my name is Ben Brakel and I'm a second-year medical student at UBC. Today I'll be presenting a project where we've used functional genomic screening to uncover drivers of survival and therapy resistance in glioblastoma stem cells. Glioblastoma is a lethal brain tumor that, despite treatment with chemoradiotherapy, inevitably recurs due to a highly dynamic population of treatment-resistant glioblastoma stem cells. Little is known about the functional drivers of glioblastoma stem cell survival and resistance to genotoxic therapies, and that's what we aim to investigate in this study. To start, we conducted genome-wide CRISPR-Cas9 knockout screens in several patient-derived GSC models as diagrammed in panel A. Gene essentiality scores are shown in panel B for 15 GBM models and 4 healthy fetal neural stem cell models, with 33 genes identified as GBM-enriched genetic dependencies. To see how these dependencies changed in response to therapy, we conducted another set of CRISPR screens under therapy exposure with radiation, temozolomide chemotherapy, or a combination as diagrammed in panel A. Among the top hit genes in panel D, we identified FEN1 as a target which became more essential upon exposure to therapy. FEN1's involvement in temozolomide-induced DNA damage repair, shown in panel E, was intriguing, and FEN1 knockout, shown in panels F and G, significantly reduced proliferation and sphere formation of glioblastoma stem cells. To determine how this dependency on FEN1 extended to therapy, we acquired a small molecule inhibitor of FEN1. This brought about two phenotypes, with healthy neural stem cells and several glioblastoma stem cell lines resistant to the inhibitor, while other GBM lines were highly sensitive as shown in panel A. When combined with chemotherapy temozolomide, these sensitive glioblastoma stem cells, which already relied on FEN1, further showed synergy between the FEN1 inhibitor and temozolomide, confirming this dependency does indeed apply to therapy resistance. Comparing a patient-matched primary and recurrent pair, only the highly aggressive recurrent GBM showed a dependency on FEN1, and furthermore this synergy was only seen in this model. To gain mechanistic insight, we found FEN1 protein levels were higher in the GBM models sensitive to FEN1 inhibition, as shown in panel A. In the patient-matched primary recurrent pair, panel B shows that FEN1 levels increased more after temozolomide treatment in the recurrent GBM compared to the primary. Exploring the DNA damage response, 3 markers of double-stranded break repair pathways showed that a DNA damage response was induced by FEN1 inhibition, specifically in the recurrent GBM model, while showing no effect on the primary model. Together, these data suggest that a subset of GBMs have a reliance on FEN1, which becomes even more important when exposed to therapy, and that inhibiting FEN1 can lead to overwhelming DNA damage and thus target these models with specificity. Our current aim is to identify why certain GBMs are more dependent on FEN1. Preliminary work has revealed that models sensitive to FEN1 inhibition and more reliant on FEN1 are more proliferative with Ki67 immunofluorescent staining and have higher DNA damage protein levels, indicating greater genetic instability. Thank you for listening, and I look forward to seeing everyone at the poster session.